Hello everyone, welcome to my lecture series on molecular dynamics and simulation. Before we begin, just read out this disclaimer. Well, most of the researchers working in statistics physics has committed suicide. Tragically, in 1906, Boltzmann committed suicide, and many believed that statistical mechanics was the cause. His student, Paul Ehrenfest, carrying on Boltzmann's work, died similarly in 1933. William James in 1909 was found dead in his room, probably due to suicide. Bridgman, the statistical physics pioneer, committed suicide in 1961. Gilbert Lewis took cyanide in 1987. And now, my friend, you have choose to select this elective subject. Of course, that was a buzzinga, and how can we discuss physics without referring this guy? Okay, I hope you know who this fellow is. Okay, well, now with this motivating pep talk, let us begin with the course of classical mechanics and molecular dynamics. By the way, I assure that no one will die learning MD simulations, at least here. So, anyways, jokes apart, today is our first lecture, and I plan to take this first session in two parts. Here, I will just introduce you to the concept of molecular simulation, why molecular simulation is very important, and what are the different type of molecular simulations that are popularly used in research community. Okay, so what my objective is that I will be giving you a bird's eye view on these simulation techniques today and in next video as well. So of course, we will dive deep into the theory and mathematical derivation in the upcoming few videos. So my main objective of this lecture and the next lecture as well is to make you aware of various simulation techniques and to brief you on as to what technique is to be used when. Okay, like when to use what type of technique. Okay, so my approach to create this video is to explain you various concepts that are of practical value when you perform these simulation in your computer or in your laboratory session. So during my career as a teacher at University of Pune, I came across a lot of postgraduate and PhD students who can effectively set up the simulation and run them as well. But they cannot somehow troubleshoot when the simulation software starts to throw those weird errors. Okay, so such thing happen due to insufficient theoretical background of this technique. So we will look behind the underlying mathematical and physical principles of various terms that are used in MD simulation. That is going to be the very core of these talks that I am going to give you in next few video lectures. I am preparing these set of videos specifically targeting the Indian student audience. Well, I tried to made an attempt that these series of videos will be beneficial to postgraduate and PhD students in Indian universities who are pursuing life sciences career or chemical sciences career. So this is what my target audience is. Okay, so now if you have a very basic biochemistry background and you are attempting to learn MD simulation, then you have landed up on the very right channel, my friends. Okay, therefore I will not assume the viewer to have any biophysics and mathematics background to understand these topics. So mathematics and physics background of 12th or even higher secondary school, that is 12th or HSC level, will be enough to start with these video lectures. Okay, So the credits are of course due. Well, during the pandemic session, I attended lecture series of Professor Juan Jose Nogueira. So he's from Department of Chemistry, Autonomous University of Madrid, Spain. I would highly recommend you to attend his lecture series that are posted on his research group's website, or you can also follow his YouTube channel. His videos assume that you have UG level. UG level, by that I mean you are at least BSc or engineering level mathematics and physics background. Okay, so that is where his lecture video begins. So if you have advanced level mathematics and physics background, I would recommend you to follow his channel. So content here might seem a bit of redundant to you. I am therefore very grateful to Dr. Juan for his wonderful lecture series that form the very basics of this video, and I am also thankful for his support he offered me in terms of fruitful discussion and providing me appropriate reading material. I would also like to express my sincere thank to Dr. Shailendra Dahivade. uh, Shailendra is my friend, and he is a faculty at Department of Physics, University of Pune. So he helped me simplifying various mathematical expression and derivation that we are going to learn in upcoming few videos. Okay, so next comes the very important question as to what should I refer after watching this video or during this video. What should I actually read? So these are some basic molecular dynamics and simulation books that might be present in your school library or in your college library. Okay, so this book will say, for example, if you are very beginner in molecular dynamics and molecular modeling, I would refer you to read this book named Molecular Modeling Principles and Application, written by Andrew Leach. Okay, similarly, you can also start out with computer simulation of liquids, written by Allen and Tilste. Okay, so these are the books that you might start out with, and then maybe after you followed all of these molecular dynamics videos that I am going to discuss, and you have some basic background on as to how things work in molecular dynamics, then you can start out reading for deeper understanding the remaining three books, like Understanding Molecular Simulation by Frankel and Smith, and you can also read this Art of Molecular Dynamics Simulation by Rappaport, and Molecular Dynamics Simulation Fundamentals and Application by Zhou and Liu. So these three books would be sufficient for you to start out somewhere. Okay, so that would be a wonderful start for you, right? And do also try to download and read a few paper that I cite in the corner of slide over here. Okay, so keep track of those references as well. If possible, download those papers and read. It will help you understand what is exactly going on in this lecture, or if you want to dive deep into details of these concepts. Okay, so well, let us begin the part one of our series. Okay, or part one of this talk. To begin with, let us first discuss as to why is that we need to study this molecular dynamics and simulation at the very first place. Okay, so you might have studied how to visualize a model of a protein that you might have downloaded from PDB. In fact, you might also know some basic molecular modeling techniques like molecular docking, where it becomes possible for you to study protein ligand interactions. Okay, but my friends, when proteins are present in cells, or you can say at when proteins are present at physiological conditions, they no longer remain static, as they appear in PyMol or Chimera or Deep View or SPDB, wherever you might have visualized those protein molecule. Okay, what my point is that proteins no longer remain static when they are present in cells. So proteins are under constant state of dynamics. They are in continuous state of motion. Say for example, some local region of protein like flexible loop may move and enable it to achieve open or closed state, right? Well, say for example, there might be say some loops in your proteins. It can be something like this, and these loops can flap around and decide if your protein is in open or closed state. So usually such open or closed states decides if the protein are in active or in inactive states. Okay, so there might be allosteric changes imparted on protein that affects the ligand binding. Okay, there might be certain G protein receptor, that those G protein receptor change the conformation and control the signaling in the cells by recruiting effector proteins, right? So well, say for example, if this is the membrane, then G proteins usually G proteins receptors are usually three domain protein, and they have got such three domains. This is the domain that is present in the membrane, while this particular domain is present in intracellular portion. So what happens is that some ligand might go and attach to this protein that would impart some structural changes that are structural changes in this cytosolic portion. Okay, and this cytosolic portion then might recruit some another effector protein, which might in turn send the signal down into nucleus, wherein it might affect in transcription of some gene or shutting down some genes, say for example. So all in all, what I want to say is that these G protein receptor change the conformation and control the signaling events in the cell by recruiting such effector protein. So now whatever movement or conformational changes that can happen in your protein, they can be local. Okay, we can say that the dynamic changes are local when it involves very small region of protein in the form of loops, or if only the side chain rotates, we can call such movement as local movements. Okay. On the other hand, the movement can encompass entire global rearrangement involving even domain rearrangements as such. Even domains can rearrange over each other. Okay. So what I want to convey is that your movements in the protein can be local, as I have discussed over here. That kind of involve movement in just small region of your protein, or it can involve very dynamic rearrangement of the protein, which can eventually encompass global rearrangements and even domain rearrangements. Okay, so the scale can be different. Okay, so now it is even believed that this motion in protein imparts functionality to it. Okay, so in short, dynamics or conformational changes in protein usually play a very crucial role in protein physiology, and therefore it is very important to study such motions that happen in protein that eventually make cells function properly. So that is why, my friend, it is very important that you study how the protein moves, what movement happen in your proteins, and how is that those movement make your protein functionally very active. Okay, so now let us talk about as to what are the experimental techniques that are proposed for that are proposed for proposed for following such dynamics in the proteins as such. Okay, now this protein dynamics can span for several order in magnitude of time as well as space in which such motion happens. So by that what I mean is that the magnitude of such motion can vary. Okay, so such motion can take place in just a flick of time, flick of second, even flick of nanosecond you can say, and or it can take a lot of time to happen. Okay, so such conformational changes in protein can happen on small region of your protein. Okay, or it can encompass a very big chunk of your protein that might involve in such motions. Okay, so in short, for example, you can say atoms in protein can move in a range of angstrom to even nanometer. Well, that is what I want to say. This atom in your protein can move from one place to another place in your protein, and this length can be in terms of, well, say for example angstrom, or they can also move in a magnitude of say some nanometer. Okay, so this is the range in which the proteins are known to move. Okay, the conformational changes that can happen, right? So if you are considering the time that is required for motion, then you can categorize them into fast motion and the another category that can be called as slow motion. Okay, so in these fast motions, such fast motion will happen in the range of even femtosecond to nanosecond. Okay, so now such motions usually comprise of atomic motion. Okay, this atomic motion can include the vibration of atoms in bond, angle bending, and even side chain flips can also be included in such fast motion that happen in the range of femtosecond to even nanosecond. Okay, now in this slow motion category, you would consider collective motions of many atoms that spans a larger area. Okay, for example, rearrangements of secondary and tertiary structural elements can even take microseconds to milliseconds of time span. Okay, so this happens in a range of even microsecond to millisecond of time span. Okay, so while such large scale domain rearrangement can take time that can range up to several millisecond to even seconds. Okay, so you can consider this paper for more details on as to how fast protein can fold. Okay, this is a wonderful paper that was published in Science in 2011, right? So now by this discussion all I want to convey is that motions can be categorized for our sake of simplicity, for our sake of studying. We can classify them as fast motion, we can classify them as slow motion. Fast motions happens very fast in scale of femtosecond to nanosecond, and they usually involve some atomic motions like vibrations and side chain flips, or even bending of certain atoms with respect to each other, while slow motions might include a larger set of atoms that would move, and that can take, well, say for example even microseconds to milliseconds. Okay, so now let us now consider as to what techniques are present that are proposed by various research group to keep track of such fast motion and slow motion. Okay, so now to track real-time dynamics, various experimental techniques are evolved. Okay, for example, you have got ultra resolution X-ray crystallography. Okay, so this ultra resolution X-ray crystallography and even X-ray laser have been used to monitor such conformational motion. Okay, so there are also techniques like ultra fast NMR. Okay, you have also got femtosecond simulated Raman spectroscopy. Okay, and this ultra fast transient infrared spectroscopy. So these technique like ultra fast NMR, femtosecond simulated Raman spectroscopy, ultra fast transient infrared spectroscopy, these are now routinely used to measure atomic motion that range from femtosecond to nanosecond. That is, if you want to monitor such fast motion, then these are the techniques that you would be interested in. On the other hand, to monitor these slow motions, there are techniques like AFM, like atomic force microscopy, that correspond to atomic force microscopy, and you also have something called as fluorescence spectroscopy, spectroscopic techniques which include fret microscopy. Fret microscopy stands for fluorescence resonance transfer energy microscopy. Okay, so using this AFM or this fluorescence resonance energy transfer microscopy, it is possible to monitor such slow motions that can span for micro or milliseconds of details. Okay, so now discussing these techniques is way out of scope of our talk. You can refer this second paper that I have mentioned over here. Okay, for more details of what are the experimental protein molecular dynamics technique that was published in scientific report just a few years ago. So if you want to go in more details of this, I would kind of advise you to read this paper. Okay, so these are all, experimental techniques that are present to monitor such structural changes that happens in your protein. Well, now all of these techniques are highly expensive. They require cutting edge infrastructure and highly trained manpower to perform such experiments. Therefore, such experimentation are not very cost effective and therefore are not frequently available in most of the university setup. So now what if we try to handle this problem computationally? Now, is it even possible to address this issue using in silico techniques? Can we find out answers to such techniques, or can we monitor such motions computationally? Well, now the answer is yes, my friends, and we can do it using statistical classical mechanics. Okay, and this is exactly what we are going to learn in the next few lecture videos. Okay, so now let us get acquainted, or let us get started with the concept behind molecular simulation method. Okay, so let me just introduce you with the concept of molecular simulation method as to what exactly do we do in such molecular simulation methods. Okay, just to begin, let us first understand the concept behind such molecular simulation, right? The basic idea in molecular simulation method is to first construct this particle based description uh, of your system. Okay, so this is what is needed to be done first. What you are supposed to do is to construct a particle based description of the system that we are interested to investigate, right? So we'll say, for example, in our case, we are interested to investigate how motions in your proteins happen. Therefore, we must first construct the particle based description of our protein. Okay, so let me tell you as to what exactly is this particle based description that we need to generate. Okay, you might have already used such particle based description of your protein. For example, to represent a protein model, we have already used this particle based description. Just recall the initial PDB visualization lecture that I have provided you. Okay, so if you remember that we have represented each and every atom in our protein in terms of its x, y, and z coordinates. Okay, so each and every atom in your protein is represented by this x, y, and z coordinate, and this particle is attached to another particle in your protein, which again have their own x, y, and z coordinate. Okay, so what I mean to say, each atom was connected to other atom using a line that represent the bond between this atom A and atom B. So this is nothing but particle based description of your protein. Okay, your protein thus correspond to a system that is made up of particle that correspond to atoms in your protein, and the lines that connect these two atoms in your protein are nothing but the bonds that connect with them. Okay, let me maybe show you the particle based description of our protein that we have kind of already discussed. Okay, so well, this was the exact 1HEW molecule that I have told you, right? So when you load this protein in visualization, this is nothing but the particle based description in which each and every atom in your protein is described by particle, and these atoms are joined by bonds, right? So over here, each nitrogen particle is described in blue color. This carbon particle is described in white color. This oxygen particle is described in red color. This sulfur particle is described in yellow color, so on and so forth. So this is what we call it as the particle description of our protein system, right? So now you have learned, right, as to how do you model a protein into particle based system. So this is nothing but your particle based system, and this is exactly what we need for molecular dynamics and simulation. Okay, or molecular simulation, say, for example. Okay, so now since you have learned how do you model a protein molecule, now someone will stand up and say ki, oh, sir, these proteins are present in water, right? Will now say you have defined your protein. Okay, now this is your protein. You learn how do we describe this protein in the particle system, right? So now someone would say ki, okay, sir, these proteins when they are present in cells, they are they are present in water system, right? Though they are always surrounded by water, so we should model those water as well. Okay, now another guy in your class would stand up and say, we also need to adjust the pH. Why? Because we know that proteins also work, proteins used to work in very narrow range of pH. Okay, another one might suggest that we need to adjust the temperature of our system as well. Why? Because at physiological condition, proteins usually work in a very narrow range of temperature as well. Okay, now let us assume that we have modeled all of this aspect. We have modeled the protein, it is surrounded by water. We have adjusted the, we'll say for example, pH in your cell by adding certain ions. Okay, and we would also somehow adjust the temperature during the simulation. Okay, now let us assume that whatever suggestion that you have assumed that we have modeled all these aspect of protein physiology. But then certainly you would realize that proteins are affected by their surroundings as well. So you might now think that you might also need to model cellular environment as well. We'll say for example, these proteins are functionally active in say some compartment. So then you might also be thinking ki, okay, I also then need to model the cellular environment like endoplasmic reticulum or Golgi complex where this protein can function, okay, that are usually present in cells. Well, then if you go on another level, cell behave on what is present in its surrounding in an organism, right? Now the surrounding is affected by climate, and climate is affected by rotation of earth, which in turn is controlled by sun, which is part of the universe. Right? The end point is that you would have to wind up modeling the entire universe. Okay, and that is not practically possible. What I mean to say is that a model is never complete and correct. Okay, so uh, what I mean to say is that model will be never complete and model would be never correct. As this wise gentleman called George E.P. Box has already stated that all models are wrong, but some are useful. Right? What he want to say is that you might never end up with a perfect model, but some of the model can be useful to draw some conclusive remarks on the studies that you are interested in. Okay, so what we usually do is that we start out with a very basic model. Okay, like the protein, in case of protein dynamics, we would start out with the very basic model of our protein. Okay, and then go on adding all the bells and whistles as and when required. Okay, so no matter what you do, the model will still remain incomplete. Well, now my question is, my friends, do you really need to model the universe in order to understand protein dynamics? Okay, so always remember that we are trying to model, what we are trying to do is we are trying to model a part of the universe. Okay, we are trying to model a part of the universe, and this part is something that is very relevant to us. So when we model something, we have to choose to leave out certain aspect and retain certain aspect of our system. So basic fact is that you should know where to stop, right? So my point is your objective my point is that your objective is to be set in a such a way that we are able to find the best answer to the given question with whatever available resources that are present around us. Okay, and in our case, our objective is to just monitor how the protein might move, right? So you should always remember this. It's true for not only biological systems but for all modeling studies. Okay, so now let me just show you uh, a simple particle based system that is generally used in molecular dynamics studies. Okay, so well, what is a typical model that we usually start up in in molecular dynamics and simulation? Okay, so let me just show you. We would be using this new software called as VMD, Visual Molecular Dynamics. Okay, so this is the new software that you might add to your list in our video lecture. Okay, so I have loaded the topology or the PSF file of this particular system, and I would have to load the data into this PSF file. Okay, I will go to this desktop. Okay, and in this desktop we have lecture 1. In this lecture 1 I'm going to load this ionized.pdb file. Load it. So now this is usually a very typical molecular system that you might want to begin with in molecular dynamics and simulation. Okay, so what you are looking at is a protein ligand system. Okay, and this this protein ligand system, which is shown in this blue color over here, it is surrounded by this water molecule. Okay, this water molecule is shown by this V. Okay, a centrally placed oxygen and the two hydrogens that are attached to it. Okay, so what you're looking at is a completely solvated box of protein. Okay, we have placed our protein is in a solvation box. Okay, because that is how the proteins are supposed to be present in cells. Okay, this protein is thus solvated in water, and we'll say we have also added some ions to we'll say, for example, neutralize the charges and to adjust the pH. Okay, so maybe I can also show you where exactly those ions are. Okay, so for that I have to see this graphics representation, and maybe I would now select the go to this selection tab, and let me see if there are ions over here. Yes, here we have. We want this ions, right? I would create a representation out of it. Okay, and maybe I will show these ions in CPK. Okay, so you can visualize if there are ions, right? Right, these are ions that are present in your CPK representation, right? So these were the ions that were present in your system. Okay, so in this way what we have is that we have this maybe I would select this protein and create a representation. Okay, and I can show this protein by lines, and these are now present in ions are now present in these spheres, right? So in this way you can see that this is a typical molecular dynamics starting point. We usually start out in molecular dynamics with building up with such systems. Okay, now let us come back to the point and say you have agreed to the extent of details you need to address in our system, right? Well, let us now agree that whatever we want to address, whatever our question is, that can be addressed using this simple system. That would we would stop at solvating our system into the water box and adjust the ion and charges, and this much would be enough. We won't need other details like its interaction with Golgi complex or the other level of abstraction. We no longer need to take care of those things. Those parameters will not affect what we are going to study. Okay, so well, let us now get satisfied. Okay, so this is the system that we are going to work with. Okay, so now we'll say once you have created the system in molecular dynamics simulation, once you have created the system, then you are supposed to, or you can say next you allow your system to propagate over some finite specified time. Okay, so well, when you propagate your system in time, we actually say that you have simulated your system for that particular time period. Okay, so this is exactly what we mean by simulation. Okay, so what we do is that we create such a system in which you have got protein that is solvated all around by water. Then you adjust the charges in your protein by adding some ions, and after your system is ready, what you do is that you would propagate your system in well, say for example, for say some time. Okay, and when you propagate your system in your time, we say that you have simulated your system. Okay, the fact is that your simulation is to be run for some limited time scale. Okay, so this is very important task. Simulations are to be run for some limited time. Okay, so you cannot continue running your simulation for indefinite or infinite time. It don't work that way, right? Your simulation should stop after some time. This is very important aspect of molecular simulation. Okay, so generally biological systems like protein ligand system that we are interested in, well, such systems are simulated over time span of say few nanoseconds to few microseconds even. Okay, so now deciding the time span is very important aspect in molecular simulation. Okay, you need to decide as to what time is sufficient for you for simulating your system so that you can answer some question. Okay, that is what I mean to say, that deciding time span of simulation is very important aspect in molecular dynamics. Okay, this time span of simulation will eventually depend on what questions you have set to answer. If your objective is to calculate the binding free energy of any ligand molecule, right? If you are trying to calculate the binding free energy of any ligand to given protein molecule, well, a simulation of say some 50 to 100 nanosecond will suffice. However, if you attempt to monitor conformational changes in protein, okay, so then you might want to simulate your system to even well, say for example microseconds. Okay, why? Because such events take longer time scale to happen, right? For calculating the binding free energy, if you simulate for 50 to 100 nanosecond, it is enough time span for you to make sure that your system is stable, and hence you can calculate this binding free energy. But if your objective is to figure out what conformational changes happen in your protein, then you might have to simulate your system for a very larger time span. Okay, you might have to simulate your system till even microseconds. Why? Because such conformational changes, well, they require longer time to happen. Okay, so now once you simulate your protein system, then you might observe how your protein system changes, or you can say what changes happen in your protein system. For example, you can monitor how hydrogen bonds forms and breaks, or you can take care of say some calculations. Well, say for example, you can monitor some values like free energy changes that might happen in your protein system. Okay, so this is how things happen in molecular dynamics and simulation, right? So now when you propagate the system for some time span, however big or small it is, you can observe how your system will evolves, or how your system changes over that period of time. By that, what I mean is that if you simulated your protein ligand system for 50 nanosecond or 100 nanosecond, right? So what you can do is that you can observe that what exactly things happen in your protein for 50 nanosecond or 100 nanosecond, or you can see how your protein evolves, or how your structure of your protein changes over the time period that you have simulated for. Okay, so now you can record these changes that are happening in your protein system. So though it is also possible to record these changes, whatever changes that happen during the simulation, and we usually record these changes in something called as trajectory. Okay, so every molecular simulation will have to generate a trajectory file, and in this trajectory file, you would record what exactly happens with your system in that particular time span. Okay, so this trajectory will show you how system has changed over time. Okay, therefore, trajectories are meant to keep track of changes that happen in your system. So in short, every simulation method must generate a trajectory that will give you an idea as to what things happen in your system, or how system changes. Okay, what things happens differently in your protein, right? So to make things more clear, well, let me show you a trajectory of a simulation. Okay, now this is going to be really very interesting, right? So now let me show you a trajectory, right? Graphics representation. Okay, and let me show this protein as in new cartoon representation, right? And let me show you this trajectory. Okay, just let me load the data from this trajectory file. Where is that lecture 1, right? And this is the trajectory file. Okay, now let it load. Now the trajectory is being loaded. Okay, now it is over. Let me close it. Extension, analysis. I will go to this RMSD trajectory tool and I would align these things. Now you can see how the protein has moved. Now you can see that there are a lot of changes that are happening in this protein, right? So protein has started to move. There are conformational changes that are happening in this region of your protein, right? So well, say for example, now you can say that the trajectory that I have loaded, it contains these 1,000 frames. Do you see? Right? So and these frames, as you can see, are shown one after another. If I click on play again, you can see that these frames are being shown one after another. Okay, and therefore it appears that this protein well, it appears like a movie as to how this protein has moved along your simulation. Okay, and this happens when you hit this play button, right? When you hit this play button, the frames in your protein will start to show one after another, and it appears like this movie. Let us talk about these frames now. Okay, so now the simulation trajectory that I have just shown you was propagated for the time span of 50 nanoseconds. So what you do in MD simulation is that you take a snapshot of your system at some desired interval of time. We call such snapshots as frames. Okay, so therefore each frame will correspond the state of the system at that particular time. Okay, for example, in the trajectory that you just saw, you can also zoom in. Okay, the trajectory that you saw, we have saved the snapshot of system after every 50 picoseconds. Okay, so let us again view this trajectory, but now I will adjust the speed of loading this trajectory one after another to very slow. Okay, and now if I hit this play button, you can see that this protein is now moving very slowly. So every time you see shift in a frame, it's the new state of protein recorded after the time span of 50 seconds. So in short, trajectory is formed by collection of frames that are freezed at some time point along the course of your simulation. Okay, to sum up, well, say for example, I can well, now you can now even see that this protein also contains a ligand, right? So let me remove this ions representation. We don't need it anymore. Okay, then let us select the residue by keyword. I know that the name of my residue is ligand, sorry. Okay, and maybe I will create a representation out of it. Okay, and I will show this ligand in, say, some van der Waals representation, or maybe I will show it in CPK representation. Now you can see let us zoom on my ligand. Okay, and now you can see that these ligand, how this ligand changes its conformation, right? So how this ligand orient itself in the active site of your protein. So say for example, this trajectory is the protein ligand complex of a flavonoid called as 7-hydroxyflavone, that is shown in this CPK representation, and the protein that you are visualizing is cyclooxygenase 2. Okay, you can see this flavonoid ring over here and how beautifully it changes, or how the atoms in your ligands are adjusted as per the protein with respect to time. Okay, so now to sum up our discussion, what we have learned so far is that every molecular simulation would end up generating a trajectory, as you are watching a trajectory over here, and each trajectory, and in this trajectory there will be different frames that are changing, right? You can see that these frames are changing. Okay, that every simulation would end up generating a trajectory. In this trajectory there will be different frames, and these frames will correspond to the state of your system at some specified time interval. In our case, shifting of every frame means that you are visualizing the state of the protein after every 50 picosecond. So this is how the protein will change its conformation after every 50 picosecond. That is what this trajectory want to convey. Okay, so this is something about your molecular dynamics and simulation. Okay, so I think we would stop at this point, and maybe in our next video I will talk about some more basic properties of molecular simulation. So thank you for your kind listening. See you guys in our next video.